Hello, everyone, and welcome to today's log reading group.
Yeah, where we are joined by Tara presenting their paper on SE3 flow matching, and we have the help of Alex Tong and Joey Bose as well. So thanks to them, and let's go.
I'm Tara. I'm a PhD student at Milan McGill, and I will be presenting our work on SE3 stochastic flow matching for protein backbone generation. And this is joint work with many co-authors, including my co-first author, Joey, who's here, and also the last author, Alex, who's, I think, also here. So, the idea is basically that using the idea of this work is to use a new class of generative models for protein backbone generation, which is called flow matching. And this ability to be able to design new proteins or de novo protein design has a lot of potential, but it's also very difficult because, it requires a lot of domain knowledge, and that's where deep learning has a lot of potential to improve the efficiency. And these proteins can be used for developing therapeutics for various diseases like COVID-19 or cancer or things like that. So that's basically what the work is about, and I will be going over some of the background material, and then go into the models themselves and some experimental results. So first, why is the paper called SE3 flow matching, and how can we represent protein backbones? So the idea is that a protein of length n has n residues, which are composed of like four heavy atoms that are connected via covalent bonds. So these are the N, C alpha, C, and O atoms. And how we can represent these proteins is to parameterize them by a collection of n orientation preserving rigid transformations, or what we call frames. So how you can do this is fix this global coordinate sort of, n star, C alpha star, etc., which are centered as C alpha being the origin. And for each of those n residues, the main atom coordinates, so the coordinates of n, C alpha, C, and O, can be seen as transformations away from these fixed coordinate system. So xi is this SE3. Xi in the bottom equation there is this element of SE3 group, which represents group of rigid transformations or rotations and translations. And we can do this for each residue. So a protein of length n with n residues can be seen as having like copies of n, as being parameterized by n frames, basically. So first, let's look at basically what this SE3 group is, and before that, what is the rotation group? Because SE3 is just rotations and translations. So SO3 is the special orthogonal group in 3 dimensions. So these are 3x3 rotation matrices, and it's a Lie group. So it has a corresponding Lie algebra or a tangent spacelet identity. And the Lie group for SO3 is the group of skew symmetric matrices, which is shown in the bottom there. And because it's a Lie group, we also have this notion of maps taking us from the Lie algebra to the Lie group and vice versa. And it's a matrix Lie group, so these maps coincide with matrix exponential and matrix logarithm. So the first map, matrix exponential, takes us from the Lie algebra to the Lie group, and the matrix log takes us from the Lie algebra to the Lie algebra to the Lie group. And we use these maps when we do flow matching for basically going back and forth between these two spaces.
Oh, and then concretely, what will be the two spaces that we're considering?
So there'll be SE3, and I'll get into that one
Okay.
second. So first, let's define a notion of distance on SO3. First, we define an inner product like you would do in Euclidean space. For SO3, the inner product is going to be in the Lie algebra. We can define it this way from the first equation, and then we also have a distance that's induced by this inner product, which is given by the bottom equation. So this is like this R1 transpose R2 is sort of equivalent to saying like the difference between the rotation matrices. R1 minus R2 kind of in the Euclidean space. And then you take the log map to go to the Lie algebra and take the norm there.
And why does
And
this like correspond to yeah, like we have one rotation matrix and another rotation matrix. And now maybe the first rotation matrix rotates like if we have our little sphere, but now we only look at like a slice and only a ring along this sphere, and then we only rotate along that ring. Now if we rotate like this and we rotate like this instead, or let's say we don't rotate at all and we just have the identity matrix and we rotate like this, and
Mhm.
then like why does this correspond to a good distance here, or what will this distance be?
So I'm kind of confused about your rotating thing, but R1 transpose R2 is sort of saying what's the rotation matrix that I need to apply to one to take me to the other rotation matrix. Does that kind of make sense? Or
So if we have our
So I think very simply, like the R1 transpose, because the group operation is matrix multiplication, and these are orthogonal matrices, right? So this is kind of corresponds to the inverse operation. So it tells you the relative rotation between the two matrices.
Okay, so this is then sort of what we would get. Like R1 transpose R2 is kind of the applying R2 and then reversing R1. And then we end up with a new rotation matrix, right? R1 transpose R2 is also a rotation matrix.
exactly. That's what and then you take the log of that.
And then you basically calculate the distance of the identity matrix to that rotation matrix. And that is
Yeah, so like that rotation matrix is basically the distance between them. So you can think about like x1 minus x2 in Euclidean space as being another element of the Euclidean space. And then you have the distance from identity to that element of the Euclidean space is basically that element itself. So
okay, let's keep going a little bit.
okay, so that's the metric on SO3. But what we're actually working in, as I said, is n copies of SE3. So we basically have the SE3 group, which is the special Euclidean group that is used to represent rigid transformations. So this group has basically, it's a 4x4 group of 4x4 matrices. It has the rotation matrix part and the translation vector. And then the Lie algebra for this is again the Lie algebra of SO3 and again it's a translation vector again because the Lie algebra of translation is just translation. So now using the metric that we defined previously for SO3, we see that this metric on SE3 sort of decomposes very nicely into an inner product on SO3 and an inner product on R3. And from that, we again get a distance that also decomposes nicely on a distance on SO3 and distance on R3. And this notion becomes important when we want to do flow matching because this allows us to sort of do flows on SO3 and R3 components separately and then just have separate distances for them and add them up in a sense.
Oh, wait.
Okay.
This is a bit weird. Like, why are you saying we can, like, we have now this square rooty thing here, where it seems to me we can't just add distances up.
Right? Oh, you have like distance squared usually, right? Anyways, so it's SO3 squared plus DR3 squared. When you, for example, have or if you have a norm, you have like basically the norm on that plus that.
Okay, so the distance can't just be added up, but
no, the distance squared is the addition of the squared distances.
Yeah.
That makes sense.
All set. And then, like, is there a simple intuition on why one matrix element disappears and in the SE3 algebra compared to the SE3 group?
What do you mean?
Okay, I'm not sure. Maybe Iman can clarify in the chat, and let's just continue.
Okay. So now let's look at this idea of Riemannian flow matching, where the idea is you want to do flow matching on a Riemannian manifold. SE3 is also a Riemannian manifold. And or SE3N actually is the manifold that we want to work with. And for that, I'm sure everyone is familiar with diffusion models, but just a very, very quick recap is the idea in diffusion models is that we want to basically reverse this Markov process that is transforming our data to noise. So this is given by the forward SC on the top. We have a drift and a diffusion term and a Wiener process. And a generative model in general, the idea is you want to be able to reverse this. You want to go from noise to data. And for diffusion models, specifically, when you do the reverse SCE, you can see that you have this term gradient log of the density at time t of x with respect to x, which is what's commonly referred to as a score function. And for diffusion models, the idea is that you want to parameterize the score function with a neural network. And when you do the reverse SCE, you sort of use this parameterized score function to follow this SCE backward in time. And that's how you generate images. Now, before diffusion models even became very popular, there were these other class of models called continuous normalizing flows, or CNFs, where the idea was that instead of the stochastic dynamics that we have in diffusion models, you have deterministic dynamics. So you have an ODE, or like a forward ODE that's transforming your data to noise. So you sort of have just the drift term. And then to reverse it, you just have the minus of the drift term to go back in the opposite direction. And that's what you would want to learn with a network. So why are diffusion models so much more popular than CNFs these days? And the reason is that they're basically they're much easier to train compared to CNFs. So this you can see by this equation, this loss equation that I have in the bottom. So it's modeling this gradient log conditional probability with a neural network, where the conditioning, you can think of it as the conditioning of the corrupted data on the clean data, or your data basically on the noise that was added in the previous step. And because everything is based on the Gaussian kernel, this is just like Gaussian convolution. It's very tractable. But in CNFs, they're much harder to train because, as you can see in this equation here, we need to simulate the ODE forward. So that's where the integral comes in. And this is problematic because when you want to backpropagate, you have to backpropagate through the solver, and that's very expensive. On the other hand, CNFs have easy inference because they're based on an ODE, and diffusion models have a much harder inference because you have to solve an STE. So we have this idea of simulation-based training for CNFs versus the simulation-free training of the diffusion models. Now, the question is, how can we make the simulation, how can we make this training of CNF simulation-free? And the idea is that you want to define this probability path, this time-dependent probability path, that is transforming your data to your prior distribution. And to generate data, you want to sort of follow it backwards from the prior to the data. And how can we do this? So we define a time-dependent diffeomorphic map called the flow, which is a solution to this ODE here. The time derivative of the flow is given by this time-dependent vector field UT. And we have some initial conditions for the ODE. And we say that this vector field UT generates the probability path if its flow pushes forward row 0 to row 1. So this is the push forward operation that's pushing forward row 0 to the probability at time t, basically, or to row 1. And that's where the idea of flow matching comes in. So the idea is to, instead of to basically learn this flow that's pushing forward your probability, and to do that you can learn this vector field UT. So you use a neural network to model this vector field UT, and that's like your flow matching loss. And the problem is we mostly don't know this rho t, and therefore we don't know the UT a lot of the times. We don't know how to compute this, so this loss a lot of the times is not tractable. So the idea of conditional flow matching is, what if we can factorize our probability distribution into simpler probability distributions? So instead of rho t, we'll look at rho t of xt conditioned on some latent z or some conditioner z, where this distribution is now easy. So if you can do this, we also have these conditional vector fields that are generating this conditional probability path, the same way that we had UT generating rho t. And the idea of conditional flow matching is now, instead of basically modeling the vector field itself, we model this conditional vector field UT of xt given z. And here, just a detail is that I have the loss of the RCFM, or the Riemannian CFM, and that's why we're doing the norm, the metric from whatever manifold we're working with. So for our case, it's the SO3 or SE3. And what's been shown is that this conditional flow matching loss has equal gradients to the full flow matching loss, and this means that optimizing for one is like optimizing for the other. So we can just use the conditional flow matching loss instead. Okay, so now I'm gonna get into the actual models themselves. So we're gonna first look at our first model, which we call FoldFlow-Base. And here the idea is that we want to do this flow matching that I described on the SE3 N manifold. And mostly in this part of the presentation, I'm going to be talking about SO3, like the losses on SO3, because the R3 part has been done previously. It's just a Euclidean part. And we had SE3N, which is N copies of SE3. So that's just adding everything N times. So what do we do to know? What do we need to do to build flows for proteins? So we need to be able to model a density over SE3N, but unfortunately we can't define a probability distribution on SE3N, which is invariant to SE3. And the reason is that R3N is non-compact, and that means that there are no R3 invariant measures on it. So what we do is...
What did just go on? Can you repeat that?
Yes. So we want to define a probability distribution on SE3N. That's invariant to SE3, right? But what we have to do, because there are no R3 invariant measures on R3N, what we have to do instead is just basically subtract the center of mass. We project into a subspace by subtracting away the center of mass, and then that becomes translation invariant. So that's all I'm saying here. And that's what we denote with SE30N, which is a subspace of SE3N, and it's a subgroup, so we can define everything that we did previously on this SE30N. So, oops. Okay. So now we need to define these conditional vector fields on SO3 to be able to do this conditional flow matching. So what we're trying to do is to parameterize UT of RT given some conditioner Z, and this vector field lies on the tangent space of SO3. So what I have here is like the homogeneous space of SO3, so a sphere, and that's what's like being used to show like represent everything. So we have R1, which is the source, so like you can think of it as a random matrix, and R0, which is the target that you want to go to, like the protein.
And sorry, can you explain a little what are the elements? Like we have SO3, we have this tangent space. Can you just and this element in SO3 is a rotation matrix, and an what is an element in the tangent space?
The element in the tangent space are the skew symmetric matrices, right? It's the Lie algebra of SO3 that we looked at.
And you had this exponential.
And identity are the skew symmetric matrices, and then at the other tangent spaces you can just parallel transport back to. You sort of go from like one tangent space to the tangent space at identity. That's where the Lie algebra is defined, and those are skew symmetric matrices.
Okay, and the exponential transform, or the exponential map brings us from the tangent. Okay.
So we have this conditional vector fields that lie on the tangent space. So you can think at each rotation matrix, there's this tangent space associated with them. And that's where the vector field that's like pointing into which direction you want to go to lies. And what we do for the FoldFlow-Base model is we fix the endpoint. So we set the conditioner to be R0, R1. And given this path that's connecting to R0 to R1, so we construct our flow basically using the most natural strategy, which is to build it using the geodesic interpolation between R0 and R1, which is this like black path between them. Given this black path, we want to find the vector field that transports us along this black line. And you can think of this idea of geodesic interpolation as being very similar to Euclidean interpolation. If you want to interpolate between 2 points like linearly, it's the same idea, just on the manifold, if that makes sense. So we want basically these vector fields that are sort of tangent to every point on this backline, connecting any source to any target. And just one small detail is that we have this exponential and log maps, and computing them involves infinite power series. So what we do in practice in our model is not use the matrix representation of the SO3 group, but the axis angle representation. But that's just like a computational detail. Okay, so what's the loss? Yes.
You actually always predict like 3 numbers and then also take the loss between these 3 numbers.
Why 3 numbers?
Like your UTR given Z are 3 numbers, I would have assumed. Like for a single SO3.
Oh, like you mean a vector, like 3?
Yeah.
Like a 3. Yeah. So yes, you can think about it that way, basically. We do the loss in the matrix space, actually. So we do the loss in the algebra space. But just the computation of RT, we do it in the axis angle space.
Okay, so wait, do you now predict three numbers and turn them into like a rotation matrix? Now, how do you parameterize your rotation matrix between which you take the losses?
What we do in practice is our network. Well, okay, what we do for proteins is actually kind of different because the prediction is not UT technically. It's the target itself, and then you compute the vector field using that target. This is sort of similar to how they do it in frame diff when they want to do diffusion. But if you were to do this, just everything, not predicting SO3, if you just had a network predicting the vector field directly, you would have to make sure that the output of the vector lies on the tangent space. So you have to project on the tangent space. So you would have to have, at the end of like when your network, whatever it output is, you project it to the tangent space. And that's where you do the computation to ensure that it's actually a correct thing. Otherwise, it's not gonna, like the output of the network is not gonna necessarily lie on the tangent space.
And do you want to get to that later, like to the prediction, or to how you actually compute your rotation matrix that you match, or do we want to get to that later?
If you have specific questions, we can talk about that now maybe because
okay, but then so what you do is you actually predict the coordinates of three atoms, so to say. Is that right?
we predict well, it's not really coordinates. It's again, like everything is represented on matrices, so you can think of like predicting the matrix for it.
No, but I thought what you just said is that for proteins that you like maybe predict one.
What I meant is we don't predict UT. We don't have UT as the output. We have R0 as the output.
Okay, so
and then
you still predict a rotation, like your like you could imagine, right, having your frame here of with the three whatevers and like three atoms of your frame. Yeah, let's say this is a frame and the three atoms of this frame, and then you just have these three coordinates, and your neural network predicts the three ground truth coordinates, and then you can align them, and then you can calculate the rotation matrix between them, and then or you can calculate yeah, I guess the rotation matrix between them, and then you can get your geodesic from the I guess it would be
so you could do everything in coordinates and like
that's not what you do.
In a covariant flow matching, we are actually doing the flow matching on the manifold itself. So all our data points are elements of this manifold that we're working with. So our proteins are already elements of SE3N, or some sort of representation of it, like maybe quaternion representation or matrix.
Yeah, I mean the coordinates would also be some sort of representation of that, right? And you could just operate in this representation and have this as the output of your neural network.
Oh, but just three numbers are not elements of SO3, right? Three number, like you could have an axis angle representation where you have both the vector and the angle of rotations, but just like if I just give you three numbers, you can't think of them as elements of SO3, really.
Why not? Why can't I say that's like a rotation vector, for example, and then yeah, calculate the corresponding rotation matrix? Why would that not be
everything can be seen as a rotation matrix when you fix this coordinate system, and when everything is like a transformation away from this coordinate system, if that makes sense. Right?
I don't really get it. Anyway, let's just get to what you actually do for proteins. So you predict X1, so to say, or R1 in this case, which are three numbers for you or not. So what is it?
Like a representation of the rotation matrix. So basically the different representation, they don't matter. Like if it's quaternion axis angle, you can just convert back and forth between them. So think of the output as a matrix, right? And then you use that.
Why? Like, that's the question. Like, what are you actually predicting? How do these people that now do flow matching on SO3 actually parameterize their rotation matrix? Yeah, you are one of the people. So how do you do?
Yeah. So correct me if I'm wrong, Alex. The output of the network is quaternion representation.
It's a rigid frame. It's a rigid frame.
Rigid frame.
What does that mean?
It's a quaternion plus a translation.
So it's a quaternion plus a translation. Then we transform the quaternion into the matrix version, into the matrix representation, and then that's how we compute our loss. We compute that matrix.
Do you compute like this Frobenius norm of the matrices? What?
When we predict, exactly. When we have the geodesic or like basically the norm on the manifold. So when we compute UT, you compute UT, you compute the output of the vector like V theta, and you basically do the log. So these are rotation, like basically those are already elements on the tangent space, and then you take them back to the tangent space at identity by parallel transporting them, and then that's where you compute your final loss. So this is the
Okay. And not so first of all, what does it mean to predict a quaternion? How do I do that? I mean, let's just say we have a single SO3, and like now, what's the output of the neural network? Is it 3 numbers, 5 numbers, or 10?
It's 4 numbers for the quaternions.
Okay, so predict 4 numbers. What do you do with those?
You convert the quaternion representation to the matrix representation. So these are just different element representations of SO3 and their formulas to convert.
But like if I have 4 numbers, then these, it sounds to me like right, for every rotation, I have 3 numbers that parameterize this rotation. In
quaternion representation, you have 4 numbers that parameterize.
Yeah, but then one of those numbers is useless, right? I can compute one of those numbers from the other 3.
I mean, yes, you can, but you need all 4 numbers because remember, quaternions can have the sign invariants. So you can convert between quaternions to SO3 elements, which is going to be, in our case, specifically rotation matrices. There's a direct conversion. And this is kind of what we leverage. So at the end, for the losses are on the matrices themselves. But to visualize, we use quaternions, and this is what the network predicts anyway. I hope that was clear.
Okay. So you're saying that there's like an additional sign that is taken care of by
well, that's inherent to the representation itself, because quaternions, remember, like SO3 has a double cover, right? And this is where the sign.
No,
so this is going to be too detailed in group theory, but just know that quaternions are an alternative representation for rotation matrices, and they have certain numerical properties that make them ideal for computation over other representations. But for our losses, because we don't want to deal with complex numbers, etc., we deal with rotation matrices. So you can convert between quaternions and rotation matrices and rotation angles, and yeah.
But like if it's just a rotation matrix, is like these matrices, are they skew symmetric?
No, the rotation matrix is not skew symmetric. When you compute the vector field using the rotation matrix, that's going to be on the tangent space of it, so that's still not skew symmetric. What's skew symmetric is when you parallel transport it to the tangent space of identity.
So are you saying there are multiple rotation matrices corresponding to one element on the tangent space?
There are multiple. We're predicting the rotation matrix itself, right?
So Alex is nodding heavily. Can you
Yeah, definitely. I mean, right. You could just keep rotating around and around, right? Like,
okay.
there's 2π in this, right? So there's always like multiple representations of every sort of of every rotation thing, right?
I mean, same with the angle. You can just keep adding 2π to the angle. It's the same.
Okay. Yeah, I'm just confused. Like, a rotation, the three numbers are enough to represent a rotation, right? So if we predict four numbers, then don't we need to constrain them in some way? Or it seems weird to me to predict four numbers and then project them all to, like, no matter what numbers you predict, you like this is the whole equivariance invariant spiel basically, right? Your neural network would have to learn that I predict these four numbers, but I can also predict these four numbers or these, and all of them will do exactly the same instead of just predicting three numbers. So now I'm confused. Why would we predict four numbers instead of three? It seems to this should make it harder to learn.
Go, Alex, go.
Well, I think it's it's just like I'm more concerned, right, not about the numbers that you're predicting, but the loss that you use, right? So if your loss is okay with, if your rotations are close, right? So one of the problems with the three numbers is sort of this weird thing where if you have like pi or almost negative pi, right? Like these are very different in the Euclidean space, but you need your loss to say, oh, these are very similar things, right? Because the rotations, up to a sort of mod 2π, right? And so as long as your loss takes care of this, then everything is okay. But of course, if you just wanted to do like a standard Frobenius norm, then you would have a lot of problems.
Okay, but can't I also just predict my three numbers, convert those to whatever representation you are using, and then take the loss in that representation?
So
Yeah,
you could,
so then
but then you would have to predict.
Wouldn't that be the best of both worlds?
Well, I think you want smooth outputs, right? So you don't want like a weird jump between pi and negative pi, right? And because like, oh, maybe if the thing rotates a little bit, then, I want my network to sort of output similar things, right? Like Lipschitz smoothness of the network would be nice, obviously.
Okay, and that you say would not be possible with any sort of conversion. Okay. Good. I don't entirely see why that is, but I can believe it, and we should continue a little bit. So there's been a lot going on in the chat, I see, but I think Joey, you've been taking care of it.
Yeah,
right?
I think we're okay on the chat. I've been answering the questions. Yes.
Okay, then we don't need to get into that. And yeah, okay, but yeah, you get into now what your actual loss is, okay?
Yeah, look at what our actual loss is. So again, for proteins, as we just talked about right now, our network itself is not outputting the vector field directly. We output the protein, like that rotation matrix, and then we compute the vector field from it. But again, the loss remains the same. So what we have here is the loss for the FoldFlow, just the SO3 component, so one residue of the FoldFlow-Base model. So the idea is first that the conditioner, for the conditional, we have this independent coupling, as I said. So it's just the product of the probability for R0 and R1, which is rho 0 and rho 1. And then we have this UT of RT given R0 and R1. And this is this is the quantity, that we have here. So this UT of R0, UT of RT given R0 and R1, as I said, is basically this vector field that's tangent to this black line. That's the geodesic interpolation. So RT is like a geodesic interpolation between R0 and R1, so like along the geodesic interpolant at time t. And this time derivative basically just corresponds to the time derivative of RT. But if you were to use just autograd to, because you could just use autograd to compute this time derivative, but that's kind of computationally expensive. So what we do is sort of exploit the geometry of the problem. So sort of how you would do it in the Euclidean space, if you have this velocity vector, how you can compute it is just to xt minus x0 divided by t, right? So we sort of do the same thing in now this SO3 space. So we know how to sort of subtract two matrices. That's what we saw before, like RT transpose RT0. And that's what this equation is saying. So log base RT of R0 is basically the same as log of RT transpose R0. It's just a more compact representation of it. And then we divide by T to get the velocity vector. So this is very similar to how you would get the velocity vector along along an interpolant in the Euclidean space. But now we just have a geodesic interpolant. So we're just on the SO3 manifold instead. But everything else is...
This log RT R0 that you write here is actually going on is that you get the two rotation matrices, take RT transpose R0, take the trace of it, take 1/2 times it, and that's the number.
No, not the trace. This is the log map that's giving us basically, it's the matrix log that I showed before.
Okay, I don't remember what the log map looked like, but okay.
So Hannes, just very quickly, so you're confusing it with the distance metric on the manifold. Well, here this is going to be a tangent vector, and the tangent vector is at RT, and we just take R0 to that tangent vector. That's all. Log map, remember, transports you from the manifold to its corresponding tangent space.
Yeah. Okay.
Yeah. So it's basically it's giving you a tangent vector that's equivalent to sort of subtracting the two matrices, and then you divide by T to get a velocity vector from them. If that makes sense. So next we look at FoldFlow-OT, which is the optimal transport version of the FoldFlow model. So what the idea there is that we so far defined these conditional probability paths, which deterministically map row 0 at the source to the row 1 at the target, but they're not necessarily the length minimizing paths. So we want to build these paths that are on average shorter, that lead to like on average shorter flows. And that means that they will have faster inference. And also empirically, they've been shown to have more stable losses during training. So we want to build these paths that are based on optimal transport. So what we have to do for that is to solve the OT problem on the Riemannian manifold. So what that problem is like this infimum over these push forward maps that are pushing forward row 0 to row 1, where you're minimizing this cost that's associated with transporting the probability mass with its push forward operation. So this cost is for us the geodesic cost, so the distance that we define on the SE3N manifold, which is just n copies of SE3, so n distances, basically. And this is a Riemannian OT problem. And we have theorems to show that this Monge map is actually exists on SE3-0N, which is a manifold that we're considering. And so basically the key ingredients to solve the Riemannian OTR. We show that the McCann interpolant for us is basically the push forward map happens to be the geodesic interpolant that we defined earlier. And we have this coupling, which is not independent coupling anymore, like we had previously. It's the optimal transport plan coupling. And in practice, what that means is we're sort of sampling a batch of data or proteins and a batch of noise, and we solve the OT by minimizing the geodesic cost, which was the SE3-0N distance, which naturally decompose into the SO3 and R3 components, and that's how we also had our data represented. And what this gives us is sort of what's the closest noise pairing to our initial data points, so that on average we get shorter flows. And again, for SE3-0N, as I said, we just add the geodesic cost n times. So what does that mean in practice? In practice, that means this is a loss for the FoldFlow-OT model. So what's changed here is, if you remember before, we had this Q of R0, R1, which was a distribution where we're taking the expectation over, which was just row 0 times row 1. Here instead, we have this optimal transport plan. And I have pi bar here instead of just pi because this is a loss for the SO3 components. So this is a projection of the optimal transport plan on SE3 on SO3 itself. But again, everything else remains the same. Your vector field is basically the same. It's just how you sample R0 and R1. That's based on the optimal transport plan, not the independent coupling anymore. And finally,
can you lose a few words or explain a little how this looks like in practice for your proteins? I mean, we have these conditional. We have these conditional distributions where we like conditioned on whatever our conditioned on the number of atoms. Yeah. how do you, and then if you, do you always have the same number of atoms in your batch?
Like the same length? Yes. The
So
same
why
length. You have the same. Yeah.
one person is shaking the head, the other is saying yes. Okay, so it's yes, I suppose.
I was shaking the head. Yeah.
We have
So
always,
that's an implementation detail, but like we ensure that this is the same length in the batch. But the OT problem is when you match noise, the same length noise to that, right? So like if
but
our
like
batch
my
will have like
question was just because if you have like now one of length 20 and the other of length 10, then I can't like do OT or I don't know what the distance then would be between these and how to match them up. Yeah, but what you basically do is you sample your noise or yeah, noise, and you sample your
Proteins, the data.
no. You sample your noise. On the one side you have your noise, on the other side you have your original data, and then you calculate the pairwise in your batch, you calculate the pairwise geodesic distances, and then you match up the noise with the data such that, or the noisy point with the data point that is the closest to it, so to say. Except that you don't just take the closest one, but you order them such that the overall distance is the smallest. Right? Wrong?
Yes, the overall distance is
No. This was wrong, right? I think you then sample according to the OT matrix.
exactly.
Yeah, okay. Good.
You match them so that based on the geodesic cost for both OSO3 and R3 component, and then you sample according to this OT plan. And that's your row 0 and row 1, or sorry, R0 and R1, and that's where, and then everything else is the same. You again compute your geodesic interpolant, you compute the vector field, etc. Everything else is the same. So
Okay,
basically,
and have you tried not training with the same length? Like, do you have a comparison of training with batches that have not the same length within them, and then with batches that have the same length within them where you can do OT, and then you did the OT?
No, that's a good question. But in our implementation, we can only, in our current implementation, we only have batches. We can only do batches that are the same length proteins.
Okay.
Okay, so that's the SO3 part. I'm sorry, the OT part. So again, the only thing that's different is how you do the sampling, not from independent coupling, but from the optimal transport plan. And finally, we have the maybe most complex one, which is the stochastic version. So FoldFlow-SFM stochastic flow matching, where the idea is that we want to replace these deterministic bridges with stochastic ones. So why we would want to do that is sort of inspired by diffusion, which works very well in high dimensions. So it's more robust. It's learning with noise, so it's more robust to it. And what that means for us in practice is that we want to replace our OD, so the first equation that we have, with an STE. So you just have like a diffusion in the Wiener process, basically. So as I said, the idea is to replace these deterministic bridges with guided stochastic bridges. And the Brownian bridge is like a stochastic process that you have pinned at the endpoints. So in Euclidean space, we can build these flows by using a reverse time Brownian bridge as the conditional flow between the points. So we have this vector field that we'll look at. So this is the sort of the velocity vector. And we have a scaled Wiener process where you're scaling by some time-dependent scaling, basically. And what's nice is that this flow is nice to sample from because you can sample from in a simulation, like this flow is, you can sample from in a simulation-free manner by just sampling from this normal distribution, which is a normal that's centered at your interpolation between the points S0 and S1. So S is just an element of the R3 group. And you have this variance that's sort of shrinking at the both ends. So you have like a narrow Gaussian at the ends, and then it's sort of like getting bigger in the middle. So that's then this is how you, like, you sample from, this is the distribution that you sample from, and then you can you don't have to like actually simulate the bridge itself. And we want to basically use a similar idea on SO3. So again, the Brownian bridge is defined very much the same way. You have the velocity vector that we've seen so far, and you have, again, the scaled Brownian motion. The Brownian motion is now on SO3, but that's basically the only difference. But the problem is that this distribution is like, basically, this is no longer simulation free. You might not know the rho t of RT given R0, R1. And what we do in our paper is to use a simulation-free approximation that closely matches this true conditional probability path. And we show that experimentally, this is basically very close to doing the SCE itself. So this probability path is given by the analog of the normal on the SO3. So this is called the XO3, or isotropic Gaussian on SO3. And you can see that it basically has like the very same like form. It has the mean, that's the geodesic interpolant. It's the same as what we saw before, the Euclidean interpolation. You have this variance that's shrinking at the ends and getting bigger in the middle. And well, it's technically like called a concentration parameter, but like basically, like you have, it has a very similar form.
Well, what's going on here? Like, this is basically just putting like three things and then saying the normal also has three things, so it's very similar, right?
Can you repeat that?
What's actually the density? Do you also have that here?
Oh, so if you know the actual density, we could just sample from that, right? So the idea is that we don't necessarily know the true probability path. So we don't know the true rho t, but we see experimentally that we can approximate it very closely with this rho t hat, which is the XO3 distribution. And what I mean by we see experimentally, I can maybe show, maybe I'll go to the next slide first and then go back, is we basically compare what happens if you simulate the SCE itself and if we sample from this distribution. So we do the simulation-free version and the simulation-based version, and where the simulation-free version is based on sampling from the distribution, the approximation that I showed, the XO3 distribution. And we see that these match very well for different values of use for the concentration parameter. So this provides a good approximation.
I just mean what this XO3 distribution is.
What is so it's similar to normal in the sense that, for example, it's closed under convolution, things like that. It's solution to the heat equation on SO3. It again has like a mean and a concentration parameter, so sort of in that sense mean and variance. So yeah, I don't know what you mean, how like to visualize it, or what do you mean distribution?
It's not just, I mean, the density, the formula.
Yeah, the formula for it has also a closed form formula. I don't have it here. I can it's in the paper though. I can bring it up. As it's kind of complicated, but it's either like an infinite sum series, or for the concentration parameter being less than 1, it has a closed form formula for it, which involves like some exponentials, things like that. I can't say the formula off.
I
don't think it's important. I think it's a detail. I think it's like something that we use, but it's like there's many works on this in the literature on what the formula is and how to compute it.
And the approximation also is like from like past literature, like pretty old literature. Right?
What you're saying is like X and then O and then 3.
XO3. So I guess IG sub.
IGSO3. Okay. Yeah. Okay.
Yeah, that makes sense. Okay. So let me just go back here just to show the loss. So again, the loss is very similar to maybe the OT loss. You have the optimal transport plan, but the difference is that now you're sampling from this distribution. So RT is sampled from RT tilde is sampled from this rho T of RT tilde given R0, R1, which is the XO3 distribution that we looked at. But that's basically the difference. And then you all obviously have an STE and not an ODE when you're doing the inference. So that's how you can do that's how you can do this stochastic version. And now we can just look at some experimental results from the three models. So just very quickly, I'm going to go over the protein metrics, which are used to evaluate the models. So the first one is designability, where the idea is that you generate 50 proteins at different lengths. And proteins have basically two representations. One is the sequence of these amino acids, and one is the structure representation, which is the representation that we use, so these frames. And what we do is we generate these proteins. We use protein MPNM, which gives like sequence from structures to get 8 sequences for structures. And then we refold them with a folding model called the SM fold. And we say that a protein is designable if it at least one of those 8 refolded structures has an SCR-MST score of less than 2. So this is the root mean squared deviation between 2 protein backbones. So this is how you measure designability, and that's what's used in the literature as well. How you measure diversity is to use the score called TM score, where the idea is that you measure basically similarity between 2 protein structures. So this is a template modeling score, I think. And yeah, it's measuring diversity. So like how far away your proteins are from each other, in a sense. And finally, we have the novelty matrix, which is also pretty important because when you want to use this for example, drug discovery, you also want novel proteins, you want new proteins. And the first one is the minimum TM score of designable generated proteins to the training data. So you can think of this as how far away your generated protein is from the training data, in a sense. And you also have a second metric, which we think is pretty important, is the fraction of proteins that are both designable. So they have this SCR-MST of less than 2 and are also novel. So for that, we use the average max TM score cutoff of 0.5, which is also used in the literature previously. So how does FoldFlow do on proteins compared to other models, which are diffusion-based mostly? So first, one thing to note, we have this horizontal line because we're comparing with frame diff retrain is the one that we can, it's the most suitable for comparison because it has the same architecture and also the same data set. The other 2 frame diff models don't really have they have slight differences in the data set. Also RF diffusion, for example, it's a much larger model, a much larger training set. It's also pre-trained, uses a pre-trained backbone and a lot of more compute resources. And I think same with Genie, the train set size is bigger, I think. Is that right, Alex? Yes, yeah. So let's look at how the models compare. So first we can see that we get the most designable proteins with the optimal transport version. So that's the one in blue. And what we see for the stochastic version that's interesting is kind of the thing that I talked about, that stochasticity helps with robustness. We sort of see that in this novelty, this fraction of designable and novel proteins. And what I mean by that is this metric in some sense sort of measuring out of distribution, generalization to out of distribution, where the distribution is our trained data. And we see that when we use the stochastic version, we get the best score on this.
So okay, in diversity, and how does the designability, so the designability gets worse with the stochasticity?
So it's sort of this trade-off between very designable proteins or just proteins that are also novel. Because if you just, you can imagine that if you just reproduce your train set, everything is super designable, but you don't have any novel proteins in a sense. So there's.
Are you using OT in SFM as well?
Yes.
Yeah, but the diversity number is the same and the designability is worse.
Yes. These are 2 different metrics. So diversity is just how diverse the generated proteins are.
Yeah, so the stochasticity is only hurting, right?
No. If you look at the novelty, so that was the point.
Novelty. Oh, okay, not diversity.
Novelty, which is, Yeah, the novelty, which is sort of measuring like, yeah, how are the proteins that you're generating actually new? Because as I said, if you just regenerate the train set, you're gonna be like perfectly designable, but definitely not novel, right?
Why do you think that is that the stochasticity improves novelty?
Yeah, that's a good question. So we think like stochasticity is helping with novelty because in some sense, it's sort of helping with out of distribution generalization. So you become more robust to noise, and we think that that's what's helping with this novelty
Wouldn't that
score.
wouldn't that mean having better designability if you're more like if you don't suffer from being out of distribution and like you're now out of distribution, so you produce a shitty structure and yeah,
What I
things
mean by out of
now.
distribution is not the full distribution. What I mean is just like the train set itself. So being able to produce things that are not just what's in your training set.
Okay, so that's even if we are out of distribution, then we are able to still produce something novel.
Yes. Yeah, because I mean your training set is not all possible proteins, right? So you don't want to reproduce your train set, because that's
Yeah,
not novel.
so in some sense, like the novelty metric is measuring like how far your proteins are from the closest training point, right? And in some sense, because of the noise in high dimension, the noise helps you navigate this space a bit better. So it helps you kind of cover the space a bit better, so you can like place points a bit further away than a deterministic map. You can think of it as like an SD improving in high dimensions as well. And there's been a lot of work in diffusion literature, at least, that shows that the SDs kind of help with this noise in high
So
dimensions.
it's kind of more like just doing high temperature sampling, so to say.
No, I think the training process is the thing. The temperature is, I don't think it helps in the inference.
Okay. What do you think what happens with your SF, like with your SFM training, you can still integrate the ODE, right? Are you integrating the ODE here?
For SFM, we do SCE integration.
Okay. So then couldn't we just check what happens if we integrate the ODE?
So like the probability flow D?
Yeah.
No, we can't. Because what actually happens is SFM, we combine sort of the drift and the score into one regression. And so you actually can't calculate an ODE from this. You need
Okay.
to sort of separately learn the score and the drift.
Yeah.
Yeah.
But then, yeah, we could like sort of ablate, um, like, is it actually giving us a meaningful variance, or are we just putting, like, just changing stuff randomly and producing some more random stuff, and then the random stuff is also further away from the stuff that we already had in
But we
training.
know that it's not random here, right?
Well,
Because it's
the designability gets worse.
no,
So,
but this fraction is not just a fraction of like novel things, so it's not just random things, it's novel and designable, if that
well,
makes
but
sense.
no, the designability gets worse. So,
Yeah, but yeah, that's true. The designability itself gets worse, but this is the fraction of novel and designable proteins. Maybe I can show this plot. So
okay.
here we have this max TM score to train set, which is like the novelty part, and we have the number of designable proteins. So this is the number of designable proteins that are also novel. So we don't measure just like if you just produce random things, that might be novel, but that's not what you want, right? You also want an actual protein. So this is what we measure. So 0.5 is the cutoff that we use, which I said is also used in literature. And you can see the SFM one, which is this, I guess you can't see my pointer, but it's like the dark red one, the one on the top.
Yeah.
That one has the most designable proteins for that are also novel up to the cutoff point. And then after that, like novelty doesn't mean anything.
Okay.
So it's not just like random matrices that are just far away from your distribution. That would not be good.
I mean, no, that's not entirely. Wait, no, designable. Yeah, designability is high. Okay, yeah, makes sense.
Yeah. Okay. So, and then a last thing that I want to mention is what we do in inference, which is a trick that we call inference annealing, where instead of following the theoretical ODE or STE for generating proteins, we use this multiplicative scaling of the velocity. So this is the IT here, and we use in practice constant times time for that. What we saw first is like just ablations. We saw that 10T works best, and that's what we use across the models. We saw that it leads to the highest designability. And so this is just only an inference, by the way. And one detail is that sort of we saw that in practice, we have this increase of norm at the end of inference, and we think this is kind of helping with that. But also at the beginning of an inference, we think this is helping. But yeah, we're looking into this inference annealing more, like different schedulings to use, how that leads to different different behaviors. But this is in practice what we saw, that like using 10T when we're doing inference in front of the vector field really helps with the designability.
Okay, so at time 0 we don't change anything, and time 1 we're very close. Like we're scaling up our the length of our vector by 10 times.
Mm-hmm.
Okay.
Exactly. And the last task that we do is called equilibrium conformation, where unlike diffusion models, basically what we're trying to show is that unlike diffusion models, flow models can start from any desamplable source distribution, and we sort of want to leverage this. So what we do is we model the equilibrium distribution of proteins given initial predicted structures from pre-trained folding models. So we do use this as like an informed prior in a sense. And then what we do is we compare to how this how this fold flow, which has an informed prior, compares to frame diff, which you're starting from a Gaussian prior, and also the fold flow RAND model, which is also again starting from just like uninformed prior. And this is basically what this plot is showing. So we're comparing the 2-Orthocene distance in angle space between generated and test samples over all residues, and also over the most flexible residue, which is residue 56. And we also look at the KL divergence at the most flexible residue. And we see that basically fold flow that's starting from an informed prior gets the best scores on these metrics. So this is just to showcase that starting from an informed prior can be useful for some protein tasks, and flow models are able to do this as opposed to Gaussian priors.
What is this informed prior that you're using here?
So this is what I said here. So we're given initial predicted structures, and we use those as that's the informed prior that we're using, and we're predicting. We're modeling the equilibrium distribution of a protein using that informed prior.
But you're equilibrium okay, so now we're no longer talking about protein generation, we're talking about structure generation.
Yeah. So this
okay.
Yeah. This is a different task. This is not the protein backbone generation that I was talking about. This is an equilibrium conformation generation. And the idea was to showcase the fact that we can do this informed prior, which can be useful for some protein tasks. There are other tasks that this can be useful for, I think, which we want to look into later.
And what sort of data set do you use here?
Oh, that's a good question. I think I have a skip slide here that I can go to. I forget. It's
I mean, I think this is not super important, right? It's like a data set that's kind of outlined in the paper. I think we take 200,000 MD frames as our initialization point or something like that. And we have
yeah, it's this
like
BPTI task.
So Frank's BPTI, and you have some like a long MD simulation for it, and as your informed prior, what do you use as the informed prior? Just a random sample from there?
No, so the idea is, right, like we have all these folding models that can give you one conformation, which is fairly good, but ideally you would like to generate lots of them. And so we use the informed prior that's basically some folding models, right? So we use, I think, AlphaFold, ESM fold, right, Omega fold, and say, yes, here's a good prior on sort of what the what it should be. And then we want to generate sort of what other structures it could be.
And how do you get the randomness into the structure? Right, you're now overfitting on BPTI. Is that right?
Yes. Yes,
Yeah,
we definitely are.
how do you get the how do we get
Add a little bit of
random?
noise, right? When we to these structures that we get, we add a little bit of noise
So
for the
your prior is the AlphaFold structure plus some Gaussian noise.
Yeah.
I see. Okay, and then you go from
noise.
to the Boltzmann distribution instead of going from Gaussian noise to the Boltzmann distribution, and we find that it helps.
Exactly.
But we probably don't want to make a statement about like a task where we generalize for this, do we? Because in the task where we generalize, it would be quite different, and like we now no longer. No, I don't know. I don't know. Maybe this is some evidence this would also work for tasks where we generalize. What do you think?
Oh, well, I
I
think
think I
like
would want more.
sorry, go ahead, Alex. Go ahead, Alex.
I would want more evidence that it generalizes than this, right? But I think, it's evidence towards that it potentially could, right? But yeah, it's not there yet.
Yeah. Okay.
Okay, so that was basically the last slide. So this slide is for questions.
Can we go back to the basically last slide because just to look at the numbers again? Okay. Good. Any questions from friends? Oh, yeah. First, thank you for the presentation and
Thanks for the
answering
questions.
my questions already. Yeah, this was very educational for me, and yeah, finally I got to know a little more about how this actually works. And yeah, any other questions from other friends of the sun?
If you can go back to the fold flows SFM, the equation that you had. So that equation is has Gaussian distribution with respect to the kernels, right? As seen in the rotations.
Sorry, you mean the XO3 distribution?
Yes, the exotic distribution.
So it's not really a Gaussian distribution. It's
Okay.
a kind of distribution on SO3. I said it's like an analog to the Gaussian distribution on it. Because again, it's a solution to heat equation and has some nice properties that the Gaussian distribution has. But it's not really Gaussian in the sense that like, I don't know, just add like, yeah, it's not really a Gaussian distribution. It's
Okay.
an
Okay. And this it also has some hidden properties as well, with respect to how it is represented when you are experimenting with it on the compute of your choice.
what do you mean hidden properties?
Yeah, hidden properties like some sort of desirable properties with the normal distribution, as
Yeah,
like being closed under convolutions and stuff.
exactly. So that's why I said it is closed under convolution. It has a nice closed form formula for the epsilon, the concentration parameter being less than 1, which allows for like fast computation of it. If you want to sample from it, that makes it pretty easy. You don't have to do this infinite series. So it has some nice properties. And the fact that you can, the fact that you can use it is because it is a solution to the heat equation. So it has those nice properties that the normal has.
Thanks. Just a last quick question. What was the hardware or the compute used to perform the experiments here?
Alex, maybe you can answer this one.
Yeah, we trained on like 4 A100s for 3-ish days, sort of yeah.
Oh, thanks.
Okay, then do I want to elucidate my understanding of quaternions and this tangent map and Lipschitzness of the quaternion parametrization further. Yeah, let's give it a short attempt. So yeah, why are we better off predicting 4 numbers instead of 3? What is your most concise and yeah, best explanation for that?
So I guess so it's not just the 4 numbers. Maybe you can think of predicting a rotation matrix also. In practice, we do quaternions for proteins, but if like for some of our toy tasks, we just I guess we just predict the tangent matrix directly. But I guess what Alex was trying to explain was like, imagine your network is outputting like 0. So like for example, in like you just have angles 0 and 355. Those are like super close actually, but they have really different values, right?
Mm-hmm.
So if you can actually have these, for example, rotation matrices, which are, for example, just think of a 2D rotation matrix like sine and cosine. So you have like everything is smooth. You don't have this jump. This will be easier for the network in a sense, and your loss also would be easier because you don't have to do like this mod thing. For example, you just have like a difference between rotation matrices, and that's like already well defined. If you have like 2 pi rotation, the matrix is going to like theta plus 2 pi is going to be the same matrix as just theta, if that makes sense.
And if I predict a rotation vector,
So we can, yeah, so we can, sorry.
if I predict a rotation vector, then I will have that issue. Saying.
If you just predict like a theta and a vector, you will have this jump in the theta in a sense.
All right, I learned a lot in this. Many questions from me. Sorry if they were too intensive and you already knew all this stuff, but I didn't. I learned a lot, and yeah, thank you to Joy Bose, to Tara, and to Alex Tong for that. And see you, friends, soon. I hope next week maybe. Bye.